Hi, good afternoon. Good morning, everyone. Welcome to CACA webinar. Today, we invited our speaker, Dr. Wang from Frontage Laboratories. He's going to give us a webinar talking about bioanalysis of liposome drugs by LCMS and MS. This is a CACA webinar series. So CACA has worked to provide career networking and award opportunities for scientists and students in the past 10 years. And also we promote the new technologies in the field of separation science. CACA is an organization. It also provides the workshop and the networking opportunities at PITCon and HPLC conferences. We typically provide a webinar in a regular basis, like every 1 month or every other month. And in the coming March, we are going to organize a dinner event at the PITCon conference. So if you happen to be at the PITCon, so please join us for the event. So we are looking forward to see you here. So in the past year, we have offered quite a few webinars. So they have been archived at CACA website. So if you are interested in, actually, you can come to visit. You log into our website, and then you can listen to the webinars that we had before. Here, we want to thank again all our generous sponsors to make the webinar available to us. So today, we invited Dr. Wang to give us this talk on the liposome drug analysis. Dr. Wang is the Associate Director, Senior Scientist of Bioanalytical and Biological Services at Frontage Laboratories. He has led more than 100 bioanalytical projects related to the development of pharmaceutical products. His current research focuses is in the area of bioanalytical method development and validation, that including validation of small molecule drugs and metabolites, as well as large molecule drugs and biomarkers such as development and validation for liposome or nanoparticle encapsulated drugs. His research interest also involves developing bioanalytical methods using novel techniques such as dry blood spot sampling, microsampling, and analyte derivatization. Dr. Wang earned a PhD in chemistry from Rutgers University. He received a bachelor in chemistry from Nanjing University in China. So far, he has published various peer-reviewed articles and book chapters on physical organic chemistry and bioanalytical chemistry. He is also an associate editor for Journal of Applied Bioanalysis. So here, actually, we are very honored to invite Dr. Wang to give us a presentation today. So now I'm going to hand over the screen to Dr. Wang so he can start his talk.
Okay, thank you so much, Dr. He, for your introduction. My name is Kai. I'm working at the Bio-8 department here at Frontage. So today I'm going to talk about the bioanalysis of liposome drug products by LCMS-MS. So here's a brief outline of today's agenda. I'm going to briefly talk about the history, introduction, application, and regulatory considerations for the liposome drugs. I'll also talk about the bioanalytical challenges and approaches. I'll go into detail with a case study, and in the end, there will be summary and Q&A sections. So liposomes. Liposomes were first described in the 1960s by Benham and co-workers. As you can see on the left side of the picture, liposome actually composed of a phospholipids bilayer and aqueous core. So even though there's a lot of publications regarding the applications of these liposome drugs, the literature describing the individual concentration profiles for the Encap and free drugs are still extremely sparse. So there are several different classifications of liposomes. If characterized by the size, as you can see in the left picture, there will be small liposome, a large liposome, and giant liposome. So the size of these structures range from 20 to 100 nanometer for the small structure, greater than 100 nanometer for the large structure assembly. For the giant, the liposome structures, a lot of times that the structure is greater than 1,000 nanometers. Also, if we go by the number of layers, if you look at the right-hand side, there will be unilaminar, there's multilaminar, and there's oligolaminars. So also, different liposome products can be different in charges. There will be most common is neutral liposomes. There will be also cationic liposomes. For example, you can add the DOTAP hydrochloride, which makes the liposome structure a positive charged structure assembly. So this will make the liposome easier to communicate with negative charge the analytes such as the DNA, nucleic acid. Also, there will be anionic liposome structures. So this can be achieved by adding some the fatty acids such as the oleic acid. So in this case, the liposome will be able to interact with the cell membrane. So why liposomes? Liposomes actually have a lot of applications. It can be used as carriers for drug delivery for both hydrophobic and hydrophilic drugs. It can be also used to encapsulate and protect the therapeutic agents from degradation and also improve the solubility. Number 3 is that it can be used to control the release rate. This is very important from a BA and pharmacokinetic point of view. It also can be used to facilitate the on-target delivery, reduce the toxicity for certain drugs. So currently, there are actually a lot of drugs in development during the clinical phase trials. For example, the first one, LEP-ETU, is a liposome paclitaxel drug product currently in clinical phase 1 and 2 trials. This was it has been developed by Insys Therapeutic company. The second one is called Thermodox, which is a liposome doxorubicin product currently in phase 2 and phase 3 studies. So this one is actually a thermosensitive liposome doxorubicin product. The third one, MM399, which is also called Onamide, is already being approved product. It's a liposome irinotecan product. This was approved back in 2015. There was also SPI 077, INX-0076, Pelmaquine, Lipoquine. These are the liposome cisplatin, topotecan, ciprofloxacin liposome drug products. These are currently all in clinical phase trials. So, what interests to us as a bioanalytical scientist is that the liposome actually changes the pharmacokinetics. So also the regulation agencies are aware of this. Therefore, they make additional requests for the PK results. So let's just briefly take a look at the global guidance regarding this liposome drug product development. First is the FDA guidance. So FDA actually finalized the liposome drug product development guidance back in April 2018. So the number one, they they emphasize that because of the complexity of the interaction between the drug release from the liposome drug product and the tissue uptake of the drug substance, a simple measurement of the total drug concentration is not enough. Number two is that the major metabolite which is associated with the therapeutic agent should be measured. I think this not only applies to liposome drug products, it actually applies to all the drug products that's currently being developed. The third one, as for the bioanalytical method, of course, we should have validated BA methods to evaluate both the total drug concentration as well as the free drug concentration. So this free drug actually refers to the drug that's being released from the liposome drug assembly. So I'll show you more detail in a later slide. So for the EMA guidance 2011, so it's very similar to the FDA guidance. It do pointed out that validated methods should be used to determine the concentration of the active substance in biological samples. Also, it pointed out that the concentration of the encapsulated and free drug should be determined at each sampling time point. For the Japan 2016 guidance, comparing with the FDA guidance and the EMA guidance, it actually talked about the tissue samples. So it requests the total concentration of the drug should be measured. On the other hand, they acknowledge the difficulty to measure the free and encapsulated drug in the tissue samples because a lot of times when we process the tissue samples, we use a homogenization, and this process will be very likely to rupture the liposome structures and release the free drug. So for us as a bioanalytical scientist, we should have three different methods to be developed and validated to support the clinical studies with respect to the liposome drug products. So that three methods will be used to measure the total drug concentration and encapsulated drug concentration, as well as the free drug concentration and metabolite. So here you can see that metabolite concentration is being characterized into the free drug method, and that's because the drug can only be metabolized after it is released from the liposome drug assembly. So on the top part of this slide, you can see that for the total drug we're talking about here include all the drugs, which is the encapsulated drugs and non-encapsulated drugs. And here, the encapsulated is specifically referred to the drugs that's being bounded into the liposome drug assembly. On the other hand, for the free drug, we're referring to both of the actual free drug and the protein-bound drug. So here at Frontage, we have quite an extensive experience for the liposome drug quantification under the regulated settings. So for plasma methods, we had experience working with human plasma, rat plasma, mouse plasma for all three different kinds of methods, including the total drug concentration method, encapsulated drug concentration method, as well as the free drug concentration method. For the tissue samples, we used to work with the rat tissues, fairly common tissues such as heart, rat spleen, skin, kidney, lung, liver, bone marrow, and testis. As for the mouse tissues, in addition to the common tissue, we actually worked with the tumor tissue. How to measure the total drug concentration? So the total drug concentration is actually fairly straightforward and easy. So there are two steps. The first one is to bursting the liposomes. This can be achieved by using organic solvents such as methanol, acetyl nitrile, very typical organic solvents. This also can be achieved by using the surfactants such as Twin 20, Twin 80, but a lot of times these are not quite recommended because the Twin poses a serious contamination to the mass spec. Also, we can change the pH by adding acid or bases. These are very effective for the pH-sensitive liposome structures. Also, temperature and mechanical shaking also can be used to bursting the liposomes. The second step it will be very straightforward. We will use the protein precipitation, liquid extraction, or solid phase extraction, etc., to clean up the samples before you shoot into the mass spec. On the other hand, for the encapsulated and free drug concentration measurement, the key is how to separate the free drug from the encapsulated drug. There's a major challenge here will be the instability of the liposome during the bioanalysis process. So this process is not including the extraction portion, but also the portion, the time frame between the sample collection until the sample receiving, as well as the sample processing. So the liposome are very fragile. It can be affected by the freeze-thaw effects, mechanical shaking, temperature, environmental pH, and sometimes non-isotonic conditions. So for the first one, the effective of freeze and thawing, what we usually do is that we add a cryoprotectant such as the glucose, sucrose, because these sugar molecules are able to provide some hydrogen bonding with the liposome structure assembly in order to stabilize these structures. Also, for the mechanical shaking part, we add the anti-shaking product. So these, a lot of times, it's the same as the cryoprotectant. So for temperature sensitive liposome structure, we have to strictly control the temperature. And regarding the pH, environmental pH, we also need to control the pH if the liposome is very sensitive to the pH change. Non-isotonic conditions, this is should be avoided by changing the buffer concentration, and we should control this strictly if the liposome is very sensitive to the non-isotonic conditions. So here the one very important message is that the liposome actually, when it ruptures, it affects the free drug concentration much more than the encapsulated drug concentration. This is because a lot of times in vivo samples, it contains most of the drug actually stays in the encapsulated form, and only a very tiny portion of the drug stays as free. For example, 99% of the drug stays encapsulated, while 1% of the drug stays as free. And during the sample processing, 1% of the liposome ruptures, and it will yield 98% of the encap plus 2% of the free. During this process, the encapsulated drug concentration changed from 99% to 98%. So this kind of change is very minimum and hardly can be detected using a Bio-A method. However, for the free drug, it will be doubled from 1% to 2%. This is a drastic change. So there's generally two techniques that we're currently using to separate the free drug from the encapsulated drug. The first one is called a solid phase extraction. The second one is we use certain membranes. It could be molecular weight cutoff, or size exclusion, or the ultrafiltration device. So here I show you two pictures. The left side is a typical carbon-18 SP cartridge with 10 milligram of sorbent bed and 1 cc volume. On the right-hand side is a 30K century-free ultrafiltration device. So both of these we used them to achieve the separation of the free drug from the encapsulated drug. So here's a brief protocol how to use the ultrafiltration device. So basically, for the study sample, which is a plasma containing the liposome and the free drug, as well as the metabolite, this will be loaded onto the ultra-free centrifuge device. And excuse me. So it will be centrifuged at 1500 G for 15 minutes. So fairly slow, not vigorously centrifuge. And after you collect the ultrafiltrate, it will be processed and analyzed for the free drug and the metabolite. On the other hand, for the solid phase extraction, here's a brief protocol. So basically, this SP column should be preconditioned with organic solvent first, and then typically with water. Sometimes for the liposome drug product that's very sensitive to non-isotonic conditions, we add a proteinaceous reagent. So after the column SP column is conditioned, you add the plasma samples and load it onto the SP cartridge. After this step is achieved, you will have this two portions. The first portion we call it fraction A. It contains the encapsulated drug. So this can be achieved by wash off the encapsulated drug by water or proteinaceous buffer. And then the rest of the step is fairly straightforward. You just break the liposome and analyze the liposome encapsulated drug concentration using LC-MS. On the right-hand side, you have a fraction B, which contains the free drug and metabolite. So this one, after you load the sample onto the SP column, the free drug and metabolite will be attached onto the sorbent bed. So after the encapsulated is being washed off, you will again use wash and elute with organic solvent to obtain the free drug and metabolite, and further analyzed by the LC-MS. So here I'll go into a detail with a case study to talk about how we develop and validate a liposome drug A. So for this drug, specific drug, we develop and validate three methods to quantitate the total drug concentration, encapsulated drug concentration, and free drug concentration in human plasma. So for each method, we actually had the stable isotope label internal standard to track and support the assay. So for plasma sample, we actually carefully selected and chose the glucose as a cryoprotectant. So this selection should be done prior to you move forward with the stability testings. So for the sample preparation, so the first step is to prepare the alpha-D glucose solution. This, for example, to prepare 10 grams of the 55% of alpha-D glucose solution in water, you will dissolve 5.5 grams of alpha-D glucose into 4.5 ml of purified water. So this, as you can see, is very concentrated, and it could take hours before you can achieve the viscous solution. So for the blank matrix, you add the 5 milliliters of 55% of alpha-D glucose into the 50 ml of the human plasma. So the final glucose concentration will be 5% in the blank matrix. So as for the QC preparation, you should use the serial dilution directly from the liposome drug product A, and in this serial dilution process, you should not introduce any organic solvent, because organic solvent could rupture the liposome structure assembly. So there are two important notes. The first one is that in order to mimic the real study sample, we should prepare the QC samples first in raw plasma, and then add the alpha-D glucose. So, for example, on the table, in this current slide, as you can see, after you prepare all the 4 levels of QCs, the final volume would be 10 milliliter, 9 milliliter, 9 milliliter, and 10 milliliter for the dilution QC, high QC, mid QC, and low QC concentration levels. So after you prepare these QC sample, you will then add the alpha-D glucose 10%, which is 1, 0.9, 0.9, and 1 ml respectively. So the second note is when you do the preparation, you should vortex gently, because sometimes certain liposome structure is very fragile. If you vortex too much, it will break the liposome and release the free drug. So for this drug A, we actually, for the total drug concentration measurement, we used a very simple one-step protein precipitation extraction. So some people might ask, how do you ensure that the liposome are all being ruptured and you're actually measuring the the correct concentration for the total drug? This is actually a very good question. I had this question come with being asked before by a sponsor. So the answer is that for the calibration standards, we are preparing actually using the spike solutions, which only contain the free drug. On the other hand, the QC sample and the study sample are prepared using the liposome encapsulated drug. So you can only have an accepted run if you have all the liposome structure ruptured. So for the encapsulated drug method, it's also very straightforward. We actually employed the SPE separation technique. So the QC sample and study samples are being passed through the SPE, and we obtain the fraction A, which was shown a couple of slides before, and then we use organic to break the liposome and recover the free drug. So again, in this method, the calibration standards should be prepared using the spike solutions, which contains only the free drug. So free drug method is we actually use the ultrafiltration separation technique. So this protocol also very straightforward. So all the sample, including the double blind SST, QC, and calibration standard assay samples, they were spiked with internal standard, which was prepared in 5% of glucose in water, no organic. And these were added into the centrifugal filters. Centrifuge was done at 1500 G for 15 minutes. After the ultrafiltrate is collected, it will be reconstituted and injected into the LCMS-MS. So there's also two important notes for the first box. So no organic should be introduced because the organic is known to break the liposome structure. Secondly, as you can see here, for the QC samples, I had prepared 3 sets of QCs: the free drug QC, liposome drug QC, and mixed QC. So for these types of QC, I'll delve into details in the next few slides. For centrifuge, again, centrifuge speed and temperature should be strictly controlled. So what is a free drug QC? What is a liposome drug QC, and as well as a mixed QC? So free drug QC is prepared using the free drug itself in the human plasma. Liposome drug QC is prepared also in human plasma, but directly from the liposome drug products. Mixed QC actually contains both the free drug and the liposome encapsulated drugs. So a lot of times people might ask, why do you prepare the liposome QCs in the free drug method? That is because for a lot of liposome drug products, it actually contains the free drug impurities. This can range from very tiny portion, let's say 0.1% to, I would say, 5% from our experience. So the first step in order to prepare a mixed QC is to determine the free drug percentage in the liposome drug QCs. So as you can see in the equation in this slide, so the fraction is determined by the measured concentration of the free drug in the liposome QCs divided by the nominal of liposome liposome drug multiplied by 100%. So secondly, when we do the stability testing, we prepare a QC we call mixed QC. So this mixed QC contains both the free drug and encapsulated drug, and the purpose is to mimic the real study samples. So because for actual study sample, both the free drug and encapsulated drug coexist. So free drug nominal in the mixed QC concentration is calculated by contributed by two portions. The first one is contributed by the impurity of the liposome drug product, and then the second and the majority was actually by spiked in the free drug amount. So for us, during the method development and validation, both the free drug QC and the mixed QC needs to be evaluated thoroughly. So the free drug QC, the purpose is to evaluate the the method robustness, the stability of the drug itself. On the other hand, for the mixed QC, the purpose is to evaluate the stability of the liposomes. So again, in this slide, the importance of mixed QC, because in the actual study samples, the encapsulated drug and free drug coexist. So in order to mimic the study sample, we prepared a mixed QC. So free drug percentage in the mixed QC should be mimicking the actual study sample, which can be obtained from the literature or data from previous preclinical studies. So here at Frontage, we actually successfully tested the stability of liposome using the mixed QCs. So here's a series of tests that we did. So in order to evaluate the stability of the liposome, so we employed the mixed QC approach. And in this slide, as you can see, we did a total of 4 sets of experiments that includes the benchtop stability at 6 hours, freeze and thawing stability after 3 freeze-thaw cycles. Also, we did long-term stability at -20 degrees for 4 days and 76 days, respectively. So as you can see, after the mixed QC is sit on the benchtop for 6 hours at 4 degrees, the accuracy actually passed acceptance criteria, which is around 106%. On the other hand, after 3 freeze-thaw cycles, the free drug is being released from the mixed QC, and it failed the acceptance criteria, which is about 117%. So again, after long-term storage, the long-term 4 days and 76 days, the accuracy becomes approximately 150% and 300%. So this slide shows that the liposome is actually fairly unstable. So in all these 4 sets of experiments, the only one that passed acceptance criteria is that it stayed the benchtop stability for 6 hours. So because of the specialty of this liposome structure, we not only evaluate the typical stability testings such as the benchtop, freeze-thaw, and long-term, we also did a series of stability testings which mimic the shipping and potential study sample processing using the mixed QC samples. So here in this slide is one special occasion where we froze this mixed QC sample at -20 degrees for 12 hours, and then transferred to the 4 degrees for storage at approximately 1, 2, 3, and 4 days. So this is just mimicking after the clinical site, they collect the plasma sample, they first put it in the freezer and then ship it to us. For this test, we actually conducted in 4 different QC concentration levels: QC low, QC mid 1, mid 2, and QC high. In each QC concentration level, each bar correspond to the different storage days: 1, 2, 3, 4 days at 4 degrees. As you can see, for all the concentration levels, after the sample is being stored at 1 day and 2 days at 4 degrees, it all passed acceptance criteria. But for longer storage time at 4 degrees, for example, 3 days and 4 days, all the 4 QC concentration level failed, which means that the liposome is not going to be stable after 2 days of storage at 4 degrees. Another series of testing, we froze the mixed QC at -20 degrees for 12 hours, transferred to 4 degrees for 1, 2, 3, 4 days, and then we add another frozen cycle for additional 24 hours. For this series, as you can see, with a longer storage period, the liposome actually fractures more and more. And also, if you look closely for the orange 2 bars for all concentration levels, they all failed acceptance criteria. So it's about 130% and up. for the accuracy. So this shows us, after 4 days, if we added an additional 24 hours of frozen, it will further rupture the liposome structure and release the free drug. So here, after we did a series of testings, the conclusion is that the liposome is actually stable for 6 hours on benchtop at 4 degrees. However, it's not stable for long-term storage, multiple freeze and thawing. So, the question for us is how to accurately quantitate the free drug. So because according to the result, so liposome is stable for at least 6 hours after the sample preparation at 4 degrees. So at Frontage here, we actually developed a procedure which we call the on-site separation technique, just to have the clinical site separate the free drug from the encapsulated drug in order to avoid the liposome rupture. And in this case, scientifically, it should yield the most accurate data. So here's a brief outline of the protocol for the on-site separation. So basically, after the plasma sample is collected at the clinical site, it will be divided into 2 portions. The part 1 will be added with the cryoprotectant and shipping to the BA lab for the total drug concentration measurement and the encapsulated drug concentration measurement. On the other hand, for part 2, the plasma will be used to separate the free drug directly at the clinical site. So this should be done as quickly as possible, definitely within the established stability. For the current drug, product A, we had established 6 hours of stability at 4 degrees. So in the protocol, we should achieve this separation within 6 hours. So and then the free drug will be processed through SPE, and it will be retained onto the SPE cartridge. And this cartridge are pre-labeled with sample information and sent it to the BA lab for free drug concentration testings. So because there are 2 types of separation techniques, SPE and ultrafiltration. So here the message is that you can either use SPE or the ultrafiltration. Ultrafiltration. So you have to, for ultrafiltration, you have to evaluate the absorption because a lot of drug products, they have a non-specific binding to the membrane. So that's why for this drug drug A, we actually employ the SPE technique. So there's for this on-site separation technique, there's 4 very important notes. It's all based on our previous experience. So the first one is the most important one because the free drug actually are processed and separated at the clinical site. So whoever doing the separation should be well trained. This is very, very important. So the second one, because the plasma sample for the free drug separation is processed directly after the sample recover without any further freeze and thawing, so there's no need to add the cryoprotectant. As for the sample itself, for the free drug, after we collect it on SPE columns, so we actually recommend at least 2 replicates of the free drug being collected. Actually, as the more the better, because considering that sometimes we need to do further reassay as well as ISR. So the more replicates, such as 4 replicates, is also recommended. So the last but not least, because the free drug after the collection, it will be either onto the SPE column or in the ultrafiltrate portion. So the stability you determined in the plasma will not be representing the stability of the free drug anymore. So that being said, you have to re-establish all the stabilities, short-term, long-term freeze and thawing onto the SPE column or in the ultrafiltrate. So for the cryoprotectant, because we already use this on-site separation technique, is it still necessary for the liposome samples, the samples that's used for the liposome drug concentration determination? The answer is yes, because even the total and total drug measurement and encapsulated drug concentration, they are actually using the same set of plasma sample. So we still need to add a cryoprotectant. So also we did some tests, and the data shows that the cryoprotectant can actually stabilize the liposome during the shipping and mechanical shaking process. So here is the example of the testing that we've done. So the design is that we prepare two concentration levels of QCs using the encapsulated drug products. So with and without, so for this set of the testings, we add the glucose as cryoprotectant, and we vigorously vortex the samples for up to 24 hours. As you can see, the encapsulated concentration stays within 90% to 105%, which means that the liposome actually is stable up to 24 hours with the help of the alpha-D glucose. On the other hand, so without glucose, we did exactly the same set of experiments, but the result is completely different. So as you can see in this figure, that both levels of QC failed acceptance criteria after 4 hours. So meaning that without glucose, the liposomes are actually even more fragile. So this data still shows that the liposome is the alpha-D glucose is very important for the liposome structure stability. So here's a summary. So at Frontage, we developed and validated 3 methods to quantitate the total drug A, encapsulated drug A, and free drug A, respectively. So the total drug was extracted by protein precipitation technique. Encapsulated drug is separated using the solid phase extraction. Free drug was separated either using the SP or ultrafiltration. So in order to obtain the most accurate free drug concentration, the separation for the free drug should be performed at a clinical site as soon as possible. And here at Frontage, we actually successfully employed this on-site separation technique and supported a few clinical studies. And yeah, that's all for today's talk. I would like to answer any of the questions.
Sure, thank you. Thank you very much. So here I do receive a lot of questions about this topic. So okay. Now I'm going to read some of the questions. So the first question is, what type of separation technique do you recommend using to separate mRNA as free drug from the liposome? Does SPE still apply?
That's actually a very good question. So basically, for us, when we do the separation, we would usually go with the SP first, and then we would try the ultrafiltration. The reason is that a lot of times we found that the drugs actually had the non-specific binding to the membranes for the ultrafiltration device. So SPE actually is better because for the free drug, as long as the chemistry selection is correct, the free drug is able to retain onto the SP column sorbent. So this is number one. Number two is that sometimes when there's a stability problem for the drugs, if the drug is actually is being separated onto the SP column, we for certain drug product we notice that it's actually quite stable. However, if the drug stays in the ultrafiltrate, it's not too stable. So that's from the stability point of view. I would recommend that you should try the SP first, and then try the ultrafiltration technique. but again, like all these are based on experience. So this a lot of the experiments should be conducted to reach the conclusion.
Okay, great. Thank you. The next question is, when performing the ultrafiltration process, did you find that type of router, fixed versus swinging bucket used for centrifugation impacted the result? Often clinical sites are limited by their types of centrifuge. Does this pose any issue?
Yeah, that's actually a great question. So basically, we didn't found any difference between the types of buckets, but we do found that with different centrifuge speed, it will affect the result. So I would still think that it's not your results is actually not due to the difference of the buckets, but is it possible that it's the I think when you centrifuge, the different buckets give a slightly different of the centrifuging speed. You might want to try just to lower the speed a little bit to see if it helps.
Okay, great. Thank you. The next question is, when developing and optimizing the ultrafiltration method for obtaining free drug, was there optimization put in place to yield enough volume of ultrafiltrate, or did you ever find that you needed to dilute the samples with plasma, and if this dilution factor would yield free drug as DLQ?
Okay, so for us, the strict answer is no, because for the liposome drug that we have been working with, quite a lot of them are oncology drugs. So the concentrations actually quite high. So for these drugs, we only need after the ultrafiltrate is collected, we don't need to add any plasma to dilute it. We just directly take the sample, let's say 25 microliter, and do the further processing. However, another thing is that because when we do the ultrafiltration, we actually had the internal standard pre-spiked with the samples, and the IS will be going through the filter along with the analyte. So this should helping you to track all the to resolve the recovery problems. So we didn't have any experience regarding adding plasma into the ultrafiltrate, but one thing I think for one of the drug product we worked with, the drug is not quite stable in the ultrafiltrate, so we had to add some stabilizer in the ultrafiltrate in order to have it store in the long term.
Okay, thank you. I think the next question is about the sample pH, and so would the set pH and the temperature be affected by the organism that you sampled? So, for example, is there any difference between the blood sample is withdrawn from like a rat and mouse, and that will be different with blood sample withdrawn from human being? So laboratory animal and the human being application.
I think it is possible, because that's why we need to evaluate the different stability for different matrices. So from what I've seen, some drugs are stable in human, but it's not stable in mouse or rat. So I would suggest just conduct all the stability testings based on each matrix, and that should give you the most accurate result.
Okay, great, thank you. So here is a question interested in the columns that you use. So first of all, this is a great presentation from our audience. So what is the LC column used for this study in terms of the phase dimension and maybe vendor?
Yeah, so if you're referring to the drug A, so that as you can see, it should be separated from in a carbon 18 based column. So I think the column we were using is Phenomenex Max RP, and the dimension is 50 by 2.1 with 4 micron particle size. Yeah, I think that's a column that we have been using. But again, different analytes behaves differently on the columns, so you really have to choose and optimize.
Okay, great. So have you ever encountered a free drug not being stable in the ultrafiltrate, which resulted in failure of ISR?
Yes, actually, that's a great question. So we did found that certain drugs is not stable in the ultrafiltrate because ultrafiltrate it's lacking the protection for a lot of proteins. So I think the one that we have been working with, we had the not only the stability problem, also the non-specific binding problem. So we for the ultrafiltration, I don't think we failed the ISR because of the stability. But again, if your ISR is really failed due to the stability, we should carefully stabilize the analyte before we fully validate the method. So your matrix to prepare the QC sample should be using the ultrafiltrate. So if you need to add a certain acid or stabilizer or even like some surfactants, we should do it during the method development and validation phase.
Okay, great. Thank you. So here's a question. Want to know what's the potential drawbacks of your experiments?
Potential drawback. So again, for the on-site clinical separation, this approach, if this involves a lot of clinical sites, it needs extensive training. So what we had experience where certain clinical site, is very well trained and gave a very good result. On the other hand, for others, you got like really crappy separation, and we can tell that it's not because of the method; it's really because of separation. Because if you add the measured free drug is almost the same as the total drug concentration. So yeah, I think our suspicion is that it's really lack of training. So a lot of I would say for this on-site separation technique approach, so the drawback is that it needs a lot of efforts to train everyone, and especially whoever do the separation at the clinical site.
Okay, thank you very much. Well, actually here I still have questions coming in. So actually today's webinar is a very well received webinar. I think more than 100 audience participated in, and during the broadcast, and then I keep receiving the message that someone wants to join, but unfortunately our space has been full, so we cannot accommodate everyone. So here I also have a question probably I can answer is, could you possibly share the slides? So the answer is yes. So we are going to post this webinar on our website, the CAC website, and then you can just register and log in and then to play this webinar again. So because of the time limitation, so probably we have to stop here for the Q&A sessions. So if you have further questions for Dr. Wang, I suggest you can talk with Dr. Wang, and I will also happy to forward all these questions to Dr. Wang after the webinar, so that we can have a very good exchange of the ideas of the technique. And so the next part is our poll question. So the poll question is to get some feedback of this webinar. Okay. So okay. So please answer these poll questions. Okay, so here is the first poll question. What's a major contributor to instability during your liposome analysis? So please select your answers. Okay, so now I'm going to close the first poll question, and let's go to the next one. So for the second poll question, that is, okay. Okay, when separating free drug from encapsulated drug, what separation technique is preferred? Okay, great. Okay, thank you for your answers. So now I'm going to close the second poll question. Thank you very much for your feedback. Okay, finally, here I want to say thank you to everyone, to our audience to attend today's webinar, and also thank you for Dr. Wang to give us such a outstanding, excellent webinar on liposome bioanalysis of liposome drugs. So this is a very good webinar, and so because of the time limit, so we have to stop here for today, and we're really looking forward to meeting you personally during the PeterCon conference, or meeting you virtually online through the webinar. So thank you very much, and wish you a good day. And okay, so bye-bye.
Thank you.
Okay, thank you.